All right, let's go ahead and get started with the material. First, we go over boys, then we go over girls. I always like to say the plumbing of the male is more complicated than the female, but the physiology of the female is way more complicated than the male. So, like, we balance each other out, right? All right, so start with fetal development. Since we're talking about males, we're talking about testosterone, its role in fetal development. Males have an X and a Y chromosome, right? Females have two X's. One of the purposes of that Y chromosome is it has this SRY gene, sex-determined Y region. And at about 7 weeks of gestation, which is just before you leave the embryonic stage, this gene turns on. All right? And when this gene turns on, that's going to determine that you get testes instead of ovaries. All right? So it's going to determine that you get testes instead of ovaries. So you're going to have to, so it's going to start developing this gonadal ridge. Testes are going to start developing, and they're going to start producing testosterone with help from HCG, human chorionic gonadotropin. And once they start producing that, then that's going to drive the rest of your development into the male into being a male. Okay? So, I always like to tell everybody this. We all start off exactly the same. We have the same parts, the same areas. We even develop very similarly until that gene gets turned on. Okay? Then we start developing this ridge. We start developing a penis, testes, and then fully into a male sexual anatomy. All right? You're going to see this same slide when we get to the female down that pathway instead. A couple things I want to point out that I will probably point out later again. There are structures, because we start similarly but diverge pathways, that are similar in male that there are in a female. That would be the glans penis is the same structure as the clitoris in the female because they're derived from the same tissue. They become erect. They fill with blood. You rub them and it feels good. That's their purpose. That's why they exist. Okay? Scrotum. We'll talk about all that later. Scrotum. What does the scrotum hold? Testes. All right. You know what the scrotum is without testes? Labia majora. So the labia majora are the female version of what the scrotal sac is. Okay, but they just never had the testes descend into them. All right, speaking of descent of testes, so if you notice, the testes start in development right here, which is the same spot where what else is? Ovaries. So again, starting in the same spot. But you have this little piece of connective tissue called the gubernaculum. I love that name because it sounds funny. The gubernaculum will start to retract throughout development and guide those testes through the inguinal canal into what's going to become the scrotal sac, and it'll become in their final resting place, all right, in the testes. All right. If this doesn't happen, we'll get into this, but do you know what the purpose of the testes being in the scrotal sac is instead of inside your body? Temperature. Sperm. Okay, sperm survive at a few degree lower temperature than body temperature. So we have this scrotal sac to be able to hold them there. If the testes don't descend, they're within the body. They're at body temperature. What's the survival percentage for sperm then? Very low. So if they fail to descend, That condition is called cryptorchidism or cryptorchidism. All right? And those people, unless remedied, become infertile. And I don't know where exactly the distinction is on how long it takes until you have to take care of this before they are not fertile, but be aware of that disorder. All right. If testes do not develop properly, we call that hypogonadism. All right. Hypo meaning low. Gonadism means coming from the gonad. So basically, you're not producing testosterone like you should. If this happens during fetal life, so the testes don't develop or they don't develop properly and they're not producing testosterone, you develop complete female sex characteristics, even though genetically and anatomically you are a male. You'll look like a female in every other respect. So that's what if that occurs in fetal life, rare. Doesn't happen very often. Eunuchism. You ever heard of a eunuch? If you've seen Game of Thrones, who is the eunuch? If you began with a V, what's his name? Pardon? Varys, yes. He was a eunuch. Loss of testes before puberty. All right? That was a weird story. I wasn't expecting that. If that happens before puberty, right, you still have them reproducing. I mean, you guys have seen my son. He's prepubescent. So imagine an adult looking like my son. That's pretty much what that is. Doesn't develop chest hair, doesn't develop fully develop reproductive organs. Things like that. They tend to be taller, reduced muscle mass because you don't have that testosterone driving those characteristics. All right. If somebody's castrated post-puberty as an adult, then you're going to have some regression. You'll still have a normal male, but they're going to regress somewhat. So this would be like, no, that's not what it's like at all. So you just have some regression, like maybe not as much hair developing, maybe some loss of bone or muscle mass, things like that. All right. Good summary slide of things we're going to be talking about on the next few slides. So if you want just a quick glance, come back to here. I'm not going to go over this because we're covering it in all of the other slides that come afterwards. So if we'll start talking about the testes. Excuse me. Those are the primary male reproductive organ, as opposed to the primary male sexual organ. All right, there's a difference there because the primary male sexual organ would be the penis. The testes are the reproductive organ. All right, we already know about their anatomy as far as where they sit. Their function is twofold. They produce sperm, and that is the reproductive part, and they produce male sex hormones, mostly testosterone, that give males their secondary sex characteristics, hair, deep voice, muscle mass, more red blood cells, things like that. All right. I think I mentioned this in anatomy, but it's important to remember. They're held in place by, or within the scrotal sac, by this spermatic cord. All right, and I want you to differentiate the spermatic cord from the vas deferens. All right, the vas deferens is a tube that sperm are carried in. The spermatic cord is the vas deferens plus blood vessels, vein and artery, plus nerves. All right, so the spermatic cord is one unit. The vas deferens is part of that. All right, so when they do a vasectomy, this is the cord they're cutting. But they can't just go in there, slice open, and cut, right? What would happen? You'd lose your blood supply, you'd lose your nerves. So they got to pry everything apart, which I know from experience is a lot of pressure, and it doesn't feel good. And cut. Okay, so keep those two structures separate. Vas deferens is part of the spermatocord. What do the testes look like? Well, you have, you've seen this in the anatomage table. We have that tunica albuginea, which is that white fibrous covering. And, like, I don't think, that's not on your structure list, is it? I don't think so. You don't have to find that. I will strip everything away and you'll just see the testes. So that is a protective layer. Then within there, you have a bunch of these coiled structures called seminiferous tubules. They're divided into different lobules. All right, so each section of the testes has a seminiferous tubule, like a longitudinal column of them. All right, and those seminiferous tubules are where your sperm are made and where testosterone comes from. All right, so here's what it looks like. This is one tubule, a cross section. This would be another tubule, another tubule, another tubule, and so on. All right, so you have the outer portion right here, and then you have the lumen. Development happens from outside to the inside. All right, so development of sperm happens from the outside, the outer rim of the Star Wars reference. The outer rim of the seminiferous tubule, and then the sperm put here, and then they travel until they reach a point where they get out of the testes. All right? We'll talk about all this stuff when we get to the next couple slides so you can actually see the picture of it. Between each one of the tubules, we have this areas of interstitial cells. Right here, right here. I mean, they look like triangles here, but that's just a drawing. Those interstitial cells are what produce your testosterone. All right, so those are outside the tubules. Outside the tubules, you produce testosterone. Inside the tubules, you produce sperm. So if you look at this right here, these would be pre-sperm cells, development occurring this way. Even though the head of the sperm is facing outward, they're still going to swim away from there, right? They're not burrowing this way. Okay? So that picture is a little bit misleading if you're not looking at it properly. All right. When sperm are developing, and we'll talk about the process of spermatogenesis, but now we want to talk about how the tubing is organized. When they form in here, they join this network called the ready testes. All right, and a ready testes is just a network of tubes where, it just collects sperm from all the different seminiferous tubules. And then they join into this coiled epididymis, which sits on the outside of the testes. All right, it sits on the outside of the testes. And when you do a male testicular exam, and you're feeling around, which you'll have to do, you can actually feel the difference between the testes and where the epididymis is. You can feel it. And that's one thing you should be looking for because epididymis can get inflamed from time to time. We call that epididymatitis. And it's, pardon? Yes, it is. All right. When sperm enter the epididymis, I want you to consider the epididymis like the foraging center. This is where they become mature. All right, so when they leave the seminiferous tubules, they're not motile, they're not ready to fertilize, but when they get to the epididymis, it takes 4 to 6 weeks for them to get to through the epididymis. So that means it takes 4 to 6 weeks for them to fully mature into something that can potentially fertilize an egg. All right, and it does this because the epididymis is specifically designed to secrete all kinds of nutrients and growth factors to help mature that sperm. All right, so now we'll go into a little more of the plumbing. So, testes, epididymis, all right, here, all the way around the superior portion of the, what is that structure? Urinary bladder. all the way around the backside, that is the vas deferens. The other name for that is ductus deferens. You'll hear both terms used. It just doesn't sound as good to call it a ductusectomy as opposed to a vasectomy. All right, it's a muscular tube, and it's got to be muscular because that's one way we help transport those sperm. The sperm aren't swimming through that tube necessarily. They're being pushed by peristaltic contractions. Has pseudostratified epithelium, which suggests it may have cilia, but I don't know that it actually does. So don't write that down. So it goes from the epididymis all the way until it reaches the this thing called the ampulla, where the seminal vesicles are located. So if we look at right here, these little structures on either side, right here. So this would be vas deferens, seminal vesicle. That's where they technically end. All right? So what's traveling through there? What's the only thing that's traveling through there? Sperm. Okay? Once they enter into this area, it becomes what we call the ejaculatory duct. All right? Which means once they're in there, what's going to happen? Ejaculation. All right. So, when we talk about ejaculate, what forms the rest of that? All right. Some secretions come from the seminal vesicles. In fact, most of your secretions come from the seminal vesicles. They secrete an alkaline fluid, and I used to work with, there used to be an A&P instructor here who used to give this lecture, and he'd go make it a point to say, and why do they secrete an alkaline fluid? Because female reproductive tract is a hostile environment, and you got to make sure those sperms survive. Sorry, I'm spitting all over. So we got to make sure they're protected, because when they go into that hostile environment, like it's a war, it was funny the way he explained it. But it's very acidic in the vagina, so you have to have a buffer against that acid, and that's why it's an alkaline fluid. It also is because sperm survive better in an alkaline environment. All right. It also contains fructose, and that provides energy to the sperm because they need to swim. Right. So that energy comes from fructose. All right. Prostate gland. So that is right here on either side of the urethra. So once you're out of the ejaculatory ducts, you're entering into the urethra. And as you saw in anatomy, the prostate is on either side of the urethra. And there's a bunch of little holes in there, and it secretes a substance into the urethra. And that substance is a thin, milky, again, alkaline fluid that increases volume. All right, so you go with sperm, then you get seminal vesicle secretions, then you get prostate secretions. And now you're here by these little glands called the bulbal urethral glands. And the bulbal urethral glands, they will secrete a mucus-like fluid. In fact, they can secrete a mucus-like fluid even before sperm get to that area, go up into the urethra, and come out as what we call pre-ejaculate. And the purpose of that is to lubricate the glands so it's easier to insert the penis into the vagina. Okay? So those are located there. Then, once you get into here, and it comes out as ejaculate from sperm, and we'll talk all about this later on. Seminal fluid, prostate fluid, and bulbourethral fluid. Okay, and that's the whole male reproductive pathway. All right, very briefly, the scrotum. That's where the testes are held in. It is a flap of skin with hair. Eventually. And if you take away the skin and look, there's a septum, a wall between each testy. So like no matter how much you try, you can't go down there and try to make one go to one side and one go to the other. It doesn't work that way. They stay in separate sides. Part of that is so they don't get twisted around, because you have two things dangling by cords. They can get twisted around easily. If you've got blood vessels and nerves there, you can kink them and they won't work properly. All right. Surrounding them, we have this thing called the dartos muscle. And the dartos muscle is it's a very thin muscle that is surrounding the testes, and that responds to temperature. All right. So when it's too hot, they'll relax. The testes will come down. When it's cold, they contract and they shrink up. Okay? So it is a temperature regulator. And you guys already told me why we need to regulate the temperature. Pardon? These are plastinated penises. I wanted to show something besides like the cartoons, but I wasn't about to go out and look on.
Delete the browser history.
Yeah. So, we're in a Catholic institution. They might get mad. This has happened before. I'll get a call from IT going, "What's going on?" Yeah, be an anatomy and physiology instructor. The stuff you have to look up, like nobody else is going to have this stuff on their computer. All right, penis, cylindrical organ. Its job is to convey urine and convey semen. All right, so that's the only thing that should come out of the penis. If something else is coming out, you need to see the doctor. All right, through the urethra. And then it's also specialized to undergo erection, and we'll talk about that process and the purpose of an erection. And trying to explain this to my 11-year-old son was a trip. So that it can go into the vagina for intercourse. And my son's like, and then I'm like, okay, try to fit a limp noodle into a hole. I said, then try to fit like an uncooked noodle into a hole. Which one fits better? He's like, oh, okay. All right. In the anatomage table, it's kind of funny that we're bringing this up now, because the quiz that I made for you guys, one of the hardest ones that you guys had to find was the erectile tissue in the penis for the quiz that I made. So by doing that quiz, you're very well aware that there's erectile tissue, right? And now you know where it is specifically because you had to cut and move and maneuver to find it. All right, so on the dorsal side are two corpora cavernosa, and on the ventral side there is one corpus spongiosum. All right, they're also encased in this tubica albuginea, so that and that's like a tough fibrous tissue that makes sure that they only get so big. Right, they can only expand so far. All right, the corpus spongiosum expands, and that's what becomes the glans penis, more commonly known as the head of the penis. The rest of it is the shaft or body of the penis. And the glans penis specifically, I wasn't saying this to be funny. The purpose of it is it has a bunch of sensory receptors, so it feels good. If it didn't feel good to have sex, nobody would reproduce. It's kind of if you sit down and actually work your way through it, like, it's kind of gross. So if it didn't feel good, you'd be like, ew, get me away from this wet, sticky mess. All right. At the end of the glans penis, you have the external urethral orifice, which is the opening for the urethra. And then before the child comes home, usually from the hospital, they cut off what's called the prepuce or foreskin. Okay, so that started off, way back 2,000 years ago or something, as a religious tradition. Then it became a health tradition, like, okay, if you want a clean, healthy penis, you've got to cut off the foreskin so like it doesn't build up things in there, because it will, and it's nasty and dirty and it stinks. But now people are saying, well, we're a little more cultured. We can take care of it a little bit better. Let's rethink circumcision. So less people are getting circumcised nowadays than they were in the past. All right? From what I understand, if you have a prep use, your glans penis is more sensitive than if you don't have a prep use. And if you think about it, that makes sense. If you have a layer of skin covering up your glans penis and it's riding around in your underwear all day, your glands isn't rubbing up against your underwear. So but if you are circumcised and it's rubbing up against, it's going to become desensitized. So it's going to be harder to stimulate. Okay? So it's a matter of tactile stimulation at that point. All right. When I first started teaching this stuff, it was really, bless you, uncomfortable to teach. Now I'm like this old man who's talking about stuff that ancient years. All right, spermatogenesis. This, actually, can you guys tell me what this is? Any guesses if we're talking about spermatogenesis?
Sperm cell?
No. Pardon? Yeah, it's a seminiferous tubule. Remember, development happens in this direction. This is the lumen, where you'll have a bunch of these little guys ready to swim out. All right, not swim out. Bad term. They don't swim at this point. All right, so here's what happens. All the stuff that's surrounding these cells, that's sustentacular cells, otherwise known as Sertoli cells. Dr. Sertoli identified them, so he named them after himself. Sustentacular because they sustain the sperm as they're developing. That's their job. That's their whole purpose in life, is to sustain sperm as they are developing. So they're like sperm nannies. All right? These smaller cells that you see in here are spermatogenic cells. And what I mean by spermatogenic is they are destined to become sperm. All right? Way out here, we have spermatogonia. And spermatogonia is one step in the evolution of a sperm cell that undergoes mitosis. All right? Mitosis is to regenerate a cell, right? We know that. That's how sperm populations keep going, is these undergo mitosis to keep regenerating a population of potential sperm cells. But then some of those will start going down the differentiation pathway, undergo meiosis 1, undergo meiosis 2, and become sperm cells. Okay? So this process starts at puberty. All right, so if we go to the next slide, it's going in the opposite direction. Let's say this is the outside of the seminiferous tubule. This would be the lumen right here. All right, so you're differentiating this way. So up to about here is what happens before puberty. So you're making these, you have mitosis, you're creating all of these cells that will eventually become sperm cells. And then once puberty hits, you start going through this process of meiosis, and these are the days in which they occur, how long it takes. I don't expect you to know each individual phase, but know about how long it takes to create a full sperm cell that's going to leave the testes. Okay? You undergo meiosis 1. Actually, let me ask you this. How many chromosomes do we have? 23 pairs. So how many total? 46. So 23 and 23. We call that diploid. Right? 2 copies of each chromosome. One came from mom, the other came from dad. Okay? During meiosis, what we're doing is splitting that 46 into 23. Right? So we split those chromosomes apart because when fertilization happens, your chromosomes, 23, will combine with another mom's chromosomes, 23, and then you have a full complement of 46 again. So this process of undergoing meiotic divisions will make sure that you only have 23 chromosomes, and they're each going to go into individual sperm. Okay? So that the sperm can then combine with maternal 23 chromosomes. What happens if you have an extra chromosome anywhere? Bad things like Down syndrome, Turner syndrome, Klinefelter syndrome, Edwards syndrome, things like that. So you need 23 chromosomes, not any more, not any less. Okay, bad things can happen. But that's not the point of this lecture, so we're not getting into it. All right. When a sperm cell goes through this process, it has a certain structure to it. All right. The structure is this part is the head. This part is the tail. All right. And the head contains a couple different parts you need to know. You have this, where is it? This anterior head cap, all right, which contains an acrosome. And an acrosome is a part of the sperm head that's specifically designed to release enzymes and penetrate an oocyte for fertilization. All right. The posterior head cap, just behind that, anybody want to guess what's in there?
It's on the slides, so it would be cheating if I said it.
Is it?
DNA.
Okay, damn it.
No, it's not on your slide. It's in the comments if you download.
It is right here.
Oh, yeah.
Shucks. Anyway, is this TV on, by the way? Okay. So it contains that's where the DNA is contained. So when you actually get to fertilization, this acrosome goes away. You still have this posterior head cap, and the entire sperm actually goes into the egg. The entire thing. All of it. All right. This neck piece right here in the body, which is part of the tail, that contains a lot of microtubules, mitochondria. Mitochondria give the energy to supply those microtubules, because what are those microtubules going to do that make the flagella? Move so the sperm can swim. All right? Notice that the activity of stored sperm is suppressed due to acidic environment. So what does that tell you about acidic environments? Is that good for sperm? No. If you want fertilization, it's not good for sperm. Then it gets alkaline, and sperm are like, hey, this is great. And they're capable of motility within 18 to 24 hours of storage in the epididymis. How long does it take to get through the epididymis? Was it days? No. Weeks. So within about 2 days, they become motile, but it takes another 4 to 6 weeks for them to actually get out of there and do what they're supposed to do. If they get ejaculated, what happens if they don't get ejaculated? Never thought of that. What? They degrade. They say, I hate when books say this, they get resorbed by the body. Does anybody know what that really means? that means macrophages come in and eat everything. Like, so literally you're cannibalizing yourself. So when anything, even like when you have tubal pregnancies and it gets resorbed, macrophages come in and eat the leftovers. Like, yeah, it's gross to think about, right? The truth is messy.
So is the acrosome part of, like, just a specialized part of the anterior head cap, or it says it contains a proteolytic enzyme, so I'm assuming that's what helps facilitate.
Yeah, so the acrosome is a special part of that anterior head cap. The rest has other things that will help the sperm cell get in.
So the entire anterior head cap comes off. Whatever gets
In essence, yes.
Okay.
All right, let me erase some of this. All right, so when we talk about sperm, it's in the ejaculate. What's in ejaculate? We already went through the pathway. So secretions from the seminal vesicles, secretions from the prostate gland, and some secretions from the bulbourethral glands. So what you have is sperm and fluid. In fact, I read a couple different spots. Some say 5% sperm, some say 10% sperm. So between 5 and 10% sperm. About 30% of the secretions come from prostate, and about 60% of the secretions come from seminal vesicles. So most of your fluid comes from the seminal vesicles. Prostate's like, let's add a few more things. And that's what is in your ejaculate. All right? So this is what it looks like in a Petri dish. No, that is not my sample. That would be weird. This is what it looks like under a microscope. And this video right here, I provided a similar video on your Canvas webpage if you want to see what sperm look like swimming around. There's about 1 to 2 million sperm per milliliter of ejaculate. It's a lot. Anybody know how much ejaculate is usually per ejaculation? Pardon? About 5 milliliters. Yeah, anywhere between 2 and 5 milliliters. So that's from 1 to 5 million sperm. How many do you need to fertilize an egg? 1. What happens to the rest of them? They die. And they get resorbed by the female body, macrophages. All right. So that's what's in ejaculate. As it's coming out from one body, going into another body, it undergoes this process called capacitation. All right? So capacitation, by definition, is changes that occur after ejaculation that allow the sperm to actually fertilize. So one of the things that happens, I switched it. Never mind. Do any of you have in your notes, right, it says uterux?
Yes.
With an X? Okay. I found it, and I'm like, no, that's not right. That was a typo. It's supposed to be uterus. The uterus and the uterine tubes wash away any inhibitory factors. So anything that's preventing those sperm from doing their job, the uterine and fallopian tubes wash that away. I didn't mention this, did I? Part of this head, the cell membrane, is rich in cholesterol. What cholesterol does for a cell membrane is it makes it more stable, right? It makes it more stable. It makes it more rigid, more tough. So in this process of capacitation, when you have the gradual loss of cholesterol, that weakens the cell membrane. It becomes soft, pliable, easy to break open, which is important if you want to release digestive enzymes to get into a barrier. All right. It also has increased permeability to calcium, which increases flagellar activity and a lot of cell cascade stuff that allow the acrosome to do its job. We're not getting into a molecular cell biology here. Also, what you'll see is ejaculate at first will tend to be a little thicker, and then through this process of capacitation, it gets more fluid-like. All right? And it gets more fluid-like, sperm swim easier. All right? Then when it penetrates the ovum, the acrosome, remember I said it contains digestive enzymes. One is hyaluronidase, which breaks through extracellular membrane material, breaks through basement membrane material, and also proteolytic enzymes that kind of help further break that apart. All right, so fun facts about sperm. I actually spent 5 minutes looking for that picture. We talked about the first one. Maturation, full maturation occurs after ejaculation. Full maturation occurs after ejaculation. So the epididymis will mature them to a certain point. They can't actually fertilize fully because they haven't gone under that process of capacitation until after the ejaculation has occurred. So how much ejaculation? Okay, I was wrong. 2 to 6 milliliters. All right, roughly 400 million sperm. One gets there. All right. How long are they in the epididymis? Up to 1 month. All right, so it takes all that time to produce. They get to the epididymis. You're a teenager with no game. So you may get to like 21, 22. All of that dies. Okay, it doesn't get ejaculated. What sperm like? Slightly alkaline environment. We talked about that. And low temperature. Lower temperature. Okay, they don't like acidic environments, which is why they're buffered in alkaline fluid. They swim at a speed of about 1.4 millimeters per minute. I mean, none of this you don't have to know. I don't care. Like, I have another fun fact later on about ejaculate speed. And their life expectancy in the female reproductive tract or in a petri dish, as you saw, is about 1 to 2 days. That's their maximum survival. Okay? That's important to know when you're counseling your patients about fertility. Like, make sure that if you're ovulating and you don't want to get pregnant and you're tracking it, don't have sex 1 to 2 days before that, or you're going to get pregnant. Right? That is important to know. All right, the really boring part about all this, hormones, right? They don't call them hormones for nothing. Haha. All right, this is your list of hormones, and this is what they do. All right, we're going to cover these as we go through, but I want you to realize that these two, luteinizing hormone and follicle-stimulating hormone, usually you hear them in reference to females, right? But they also do have function in males. In males, luteinizing hormone stimulates testosterone secretion. Follicle-stimulating hormone stimulates Sertoli cells. What are Sertoli cells? Right, the support cells for sperm development, the nursemaids, the nannies, right? Is that what I said, nannies? Yeah. The nannies for the sperm. So it stimulates them to drive sperm development. All right, so indirectly, FSH stimulates sperm development. All right, of course, males secrete testosterone. We'll talk about what that does. Males also secrete estrogen. All right, and estrogens, I put a little note down at the bottom, they also help aid in sperm formation and maturation. They also contribute to bone and muscle growth. And they also contribute to cholesterol metabolism because ultimately testosterone, estrogen, androgens all come from cholesterol to begin with. All right, and there's an inhibitory hormone that goes back and shuts off anterior pituitary hormones. All right, so how do we control this pathway? We have what's called the HPG axis, hypothalamus, pituitary, gonadal axis. All right, I'm going to give you a trick right now. This is also in the females. They have an HPG axis too. All right, hypothalamus secretes gonadotropin releasing hormone. Luteinizing hormone and FSH are gonadotropins. So GnRH stimulates the release of LH and FSH. All right, then LH and FSH, we talked about their effects on gonads. What's the gonad in a male? Testy. Okay, I just want to mention this quick right here. What this is saying is activated G protein coupled receptor, cyclic AMP second messenger. All that means is that GnRH, LH, and FSH are non-steroid hormones. Testosterone, steroid hormone. All right, so we'll finish this. I'm going to take a break. Male sex hormones. Remember, here is a seminiferous tubule. Here's a seminiferous tubule. Here's a seminiferous tubule. These are the interstitial cells that are secreting male sex hormones. All right. They secrete androgens in general. Testosterone is the most abundant. It circulates as a specific form, but it's not until it gets to the peripheral tissues where the receptors are located that it converts to DHT, which is the active form that does all the things that you want testosterone to do. All right. We talked about what testosterone does during fetal development. At puberty, it goes through the roof and accelerates all of the things that testosterone normally does. All right, so we'll talk about some of those things in a couple slides. This is just showing you, in fact, in the adrenal glands, cholesterol is converted to an androgen. In the peripheral tissues, testosterone is converted into DHT because the peripheral tissues have this enzyme called 5-alpha reductase. It's because they have those that it converts it to that form. If you don't have that enzyme, you don't convert to that form, whether you have a receptor for it or not. Testosterone can also be converted to estradiol by peripheral tissues, and we already know that estrogens do play a role in male physiology, just not to as big of a degree. In the testes, all of this can happen. Okay, so that's what these brackets mean. All right, if testosterone is a steroid hormone, what does that mean about where its receptor is located? Do you remember? All right, steroids are what? Lipids. So does that mean they attach to a receptor on the cell membrane, or they just go right through the cell membrane? They go right through, right? So the receptor is typically in the nucleus. If it's not in the nucleus, it moves to the nucleus after binding occurs. All right? And when it moves to the nucleus, what it does is it binds to its receptor, it goes to DNA, and usually makes new mRNA to make new proteins, right? So steroid hormones work by making new proteins, protein production pathways. All right, because it's a steroid, most of your testosterone is going to be attached to proteins as it's circulating to make them soluble. If they don't attach to these proteins, they don't circulate as well. That's why only 2% of your testosterone is free testosterone or unbound to anything. Okay? Whatever testosterone is left over, in other words, what doesn't go to cells, gets converted in the liver to forms that get excreted from your body or recycled to do other things. All right. So, what does testosterone do in a male? That's not a fetus, because we already talked about that. Gives you your secondary sex characteristics. What are the secondary sex characteristics? They're all listed here. Okay? Some general things. I think I mentioned this earlier. Deep voice, thicker skin, more muscle mass, more red blood cells to supply oxygen to that bigger muscle mass. Okay? More water retention, because muscle holds more water than fat does. And like it or not, ladies, you have more fat than a male does. And it serves a purpose. All right? It's there for a reason. The reason's reproductive. In case you didn't get that. It also increases your basal metabolic rate. All right, so that's why once boys hit puberty, and they might be a little chunky before puberty, they thin right up. A lot of times. What?
I said it
It
didn't happen.
didn't happen? All right, but if you see that phenomenon, that's why that happens.
I used to play line in football in grade school.
Do what?
I was the center. I was bait.
Yeah.
It worked.
Then puberty happened?
It worked.
All right, so let's talk about erections. So this is the end. This is like our intro to the female reproductive tract, because why do you have an erection? To go in there. All right? Sexual stimulation. Now, sexual stimulation can be mental or it can be physical. All right? It can be mental or it can be physical. In either case, what it's going to stimulate is parasympathetic neurons releasing nitrous oxide within the penis. All right? What does nitrous oxide do to arteries or arterioles? Dilates. So what's going to happen to the blood flow to the penis? It's going to increase. Well, the blood vessels in the penis are leaky. All right? And because they're leaky, they're going to fill this spongy tissue in the corpora cavernosa, primarily the corpora cavernosa, and the penis is going to swell or become erect. All right? As it does that, it's going to compress the veins that surround the penis. What's the importance of compressing veins that surround the penis? What does that do? You have a lot of blood going in, but very little going out. So, whoop, you now have an erect penis. All right? So it's going to accumulate there. All right? Notice that you don't have as much. There's a little. It doesn't show it here, but you don't have as much blood going into the corpus spongiosum. All right? If you have the opportunity. Yes, I'm going to say it. Like, I always say this to you guys. Like, an erect penis, if you feel around where the corpora cavernosa are, they're going to be hard. But if you feel the corpus spongiosum, it's going to be softer because it doesn't fill as much with blood. All right? You can do this experiment. All right? Guys, you can do it anytime you want. Ladies, you need a partner. All right, so the penis swells and becomes erect. Of course, the point of that, we talked about, is to go into the female reproductive tract. What happens is, all right, you get into the reproductive act. what is the primary stimulus for getting an ejaculation to occur? Where are the primary sensory endings? In the glands penis. All right? So this is talking about the sensory. This is talking about the parasympathetic that we just talked about. Parasympathetic also stimulates the release of mucus from the bulbourethral glands. All right? So it's not just that you have to have the ejaculatory duct filling up for this. The parasympathetic can secrete that ahead of time. All right? So what happens is the sympathetic, you have two processes for ejaculation, or for the process of ejaculation. You have emission and you have ejaculation. All right? When you contract the vas deferens, when you contract the prostate, when you contract the seminal vesicles, you're putting sperm into the ejaculatory duct, you're putting fluid into the ejaculatory duct, and you're getting it ready. If you are putting all of this fluid into a tube that has limited space, what's going to happen to that tube? It's going to expand. Now, haven't you noticed in physiology that a lot of things that happen in the body are because something's expanding? Okay? So it expands. All right? When it expands, that's going to trigger a spinal reflex. So ejaculation is a spinal reflex. I can't just think, hey, I want to ejaculate and something happens. Like, it is a spinal reflex. It doesn't require higher nerve input. So when that swelling of the ejaculatory duct happens, that's going to cause contraction of the perineal muscle, and ejaculation occurs. Does that make sense? Okay. So emission is filling up the tubes. Ejaculation is making sure it gets out. If you want to put it in real terms, emission is the, and ejaculation is the. All right, so we've talked about this. After ejaculation occurs, the sympathetic nervous system constricts those arteries, so not as much blood goes in anymore. So that allows the blood to slowly trickle out, and eventually you get back to baseline, what we call a flaccid state. Flaccid state. All right, and the fun fact about ejaculation is I don't know who did this research or how they came up with it, but apparently it occurs at 28 miles an hour, which is speeding in a residential zone. All right, so
that's why that shouldn't happen.
So we'll leave it there. Take a 5-minute bio break. We'll start with the female reproductive system.
Can I ask you a quick question?
Just as soon as I guess,
It's about that. So.
as opposed to, all right, let's talk about the female reproductive system. All right, so remember, we all start off the same. What is it that drives us down a differentiation pathway? SRY. All right, if we don't have SRY, the default pathway is female. So we are all initially geared towards becoming female unless we have the SRY gene. Okay? All right, start with the ovaries. There's my stuff. Notice where the ovaries are located. What else was located there? Testes, until they descended. What allowed the testes to descend? What was that structure? Come on, it's fun to say. The gubernaculum. All right. The ovaries are held in place by ligaments. All right, we don't want the ovaries to be free floating. They have to be held in place because they have a very specific job, and what they secrete has to go to a very specific place. So they need to be held in place, and there are ligaments. This is more of an anatomy, so I'm not going to go into detail about this, but they're held in place against the wall of the pelvic brim here. Okay. They have two parts. You have an outer cortex. Okay. You notice a lot of things in anatomy have a cortex and a medulla. You have the kidney, you have the adrenal gland, you have the ovaries. All right. The medulla is a lot of connective tissue, nerves and blood vessels, lymphatics. All right. So very nutrient-rich area. The cortex from fetal development through death has what we call primordial follicles in it. Primordial follicles. And what primordial follicles are a single egg with a layer of granulosa cells surrounding them, which provide nutrients to that egg. And females are born with all the oocytes they're ever going to have. All right, females are born with all the oocytes that they're ever going to have, which, if I remember correctly, is about 2 to 3 million. All right, ovaries are also covered with connective tissue, the tunica albuginea. What else was covered by the tunica albuginea? The testes, right? So common themes here. All right, so here's an ovary. Here's an ovary. Extending to that ovary, we have these uterine tubes, also called fallopian tubes. All right, I like uterine tubes because it helps explain what they are better than fallopian. I don't remember, I used to know where fallopian comes from. But fallopian and uterine tubes are the same things. All right, the extensions, as we learned in one of the classes that were at the anatomage table, are the fimbriae that kind of cover the ovary. They're like little finger-like projections, kind of like you have a hand and you're holding the ovary. Okay, then the uterine tube would be my arm. That would make me a uterus. Okay? The wall of the uterine tube contains a ciliated mucosal layer. What that means is that they're constantly moving in one direction. What direction do you think they're moving? Away from the ovary or toward the ovary? Away from the ovary. Because when that ovary is secreted from, that's ovulation, secretion of an oocyte. When that oocyte is secreted from an ovary, we have to have a way to get it down into the uterus. All right, and those cilia help it travel along. All right, but it also has a muscle layer, so you have some peristalsis that also helps the oocyte move down the uterine tube. All right, so uterine tube, uterine tube. The rest of this is the uterus. All right, also held in place by ligaments. All right, we're not going to go into detail about that because that's more of an anatomy thing. But its job, the uterus's job, is to receive the embryo. And what's an embryo? Basically a ball of cells, right, from a fertilized egg. It receives the embryo and helps sustain it. So it's the uterus where the baby is going to develop. All right? Anatomically, I want to go over this really quickly. And I know you talked about this in clinical correlations, and you've probably studied it in anatomy. When you're looking anterior to posterior, urinary bladder, uterus, rectum. Okay, anterior to posterior. That's the way it works. Okay. The reason I say that is I don't want any of you going to an anatomy exam and labeling the urinary bladder as the uterus, or vice versa. All right, extending into the vagina. Almost sound like I was going to say vajayjay. The vaginal canal is the neck of the uterus, and the neck of the uterus is called a cervix, right? I always wondered, why do they call it a cervix? Like cervix means neck. You have cervical region, right? Neck. It's the neck of the uterus. All right, the wall of the uterus is a little bit differently than what you see in the uterine tubes. It has the endometrium, which is like a mucosal layer, all right, that builds and sheds, builds and sheds. The myometrium, which is a muscle layer that surrounds it, and the muscle layer is specifically designed for the birthing process. And the perimetrium, which surrounds it. So, many things have layers. The important ones that we're going to talk about is the endometrium. All right, so here's our uterus. We're not considering a uterine tube here. Ovary, cervix, the rest of the tube, and I think I've said this many, many times in both anatomy lectures, in lab with you guys, the vagina is the tube, not the thing you see when you do a physical exam, not the thing you first see, okay? Even though commonly that's what your patients are going to say, that, like, my vagina. Like, well, your vagina is when I stick my finger in there to do the exam. That's your vagina. What you see here, do education. Educate your patients. The purpose of a vagina is twofold. Receives the penis for intercourse. It secretes things. And it's basic, like, forget that part. Like, it does that, but really what purpose does that serve besides putting things in there or getting things out, right? So it provides a canal for birthing as well. All right, now when the vaginal canal forms, it's partially covered initially, and that's called the hymen. And if you look, these are a bunch of, they look like spoons. I never noticed that before. Anyway, so partially covered, and they have different morphologies, different appearances, and those are properly named. You don't have to know these names. I just want to give you a feel for, like, when they say there's a hymen. This doesn't look like there's one there, but it is partially closed. Okay, so that closes, and eventually that gets broken, and you have a full vaginal canal. All right, what it's made out of is a mucosa of stratified squamous epithelium. What does stratified squamous epithelium mean? Has a lot of layers to it. Why would a vagina need a lot of layers? It's getting friction, right? It's getting rubbed against, whether it be intercourse or whether it be a baby coming out. So it's got to have protective layers. That's why it's made out of stratified squamous epithelium. Layer of smooth muscle because it contracts, and a fibrous coat, okay, around the outside. Ladies, I'm going to tell you right now, this picture has always bothered me because you never see a male counterpart with the same, a male picture showing the same thing. Like, to me, that's weird, right? I mean, it's the best way to see this, but, like, to me, like, if I'm a female sitting there, I'm like, oh, I feel exposed. All right. Looking at the external anatomy, we have the labia majora here. What was the male equivalent? The scrotum, right? So those are rounded folds of adipose tissue and skin, okay, which if you're a male, just you lose the adipose tissue because you have testes in there. And the labia minora, which are these flat longitudinal folds which keep things closed. All right, at the anterior end, we have the clitoris. All right, and again, the clitoris is analogous to the penis, with the tip of it being analogous to the glans penis. And it is sensitive like the glans penis is. All right, and remember, it has a very similar structure underneath, where you have a crus that's attached to the anterior wall of the pelvic bones. And then this area here, just posterior to that, is called the vestibule. So the opening is the vestibule. All right, a pre-entry area. All right, and in that pre-entry area, two things are important. Three things. You have the vaginal opening, or the opening to the vaginal canal. You have the external urethral orifice, which opens to the urethra going to the urinary bladder, which is short relative to a male. And clinically, female urethras, difficult to find if you're talking about catheterization. I made it through all of nursing school and clinicals, never having to put a catheter in anybody, which was amazing. I don't know how I did that. I had to clean one in a male, and you guys told me that you heard that story on my lectures, but I've never actually put one in. But from what I understand from those who've done it, like, sometimes female anatomy is so different that it's hard to find where the orifice is. And a lot of times you'll get the catheter into the vaginal canal. And what happens when you do that? You got to throw it away. You got to get a new one because it's got to be sterile. All right. We also have these Bartholin's glands, which are similar to the bulbourethral glands, and their job is to convey secretions to provide lubrication, all right, for intercourse. All right. So most of this, we're going to be talking about the female reproductive cycle. So it's a lot of charts and hormones going up and down and things. I know it's boring, but we got to go through it. All right. The female reproductive cycle, also called the menstrual cycle, has two goals: release of an egg, all right, to get fertilized, and to prepare the uterus for the egg. Like, we need an egg, and it's like tilling the earth to plant seeds. All right, we got to prepare it so that it implants properly and grows. All right, so it's divided into two major phases, and actually we'll see that it subdivides: follicular phase and the luteal phase. Okay, so we'll talk about the follicular phase first, but I'll introduce it by saying, remember, we have this primordial follicle, which is an oocyte surrounded by granulosa cells. This whole thing is called a follicle. So the follicular phase would be development of that follicle. All right, so we talked about the primordial follicles, where they're all located in the cortex, the purpose of the granulosa cells. It also secretes, the oocyte secretes something that inhibits maturation until it gets a signal. Okay, and the signal overwhelms that inhibiting factor, and then you get development. All right? When it starts to develop, it's going to start to get bigger. The oocyte's going to get bigger, and the granulosa cells are going to start dividing, right? So the whole follicle is going to get bigger as well. That is regulated by follicle-stimulating hormone. So now follicle-stimulating hormone makes sense, right? It stimulates the development of a follicle, and a follicle contains the oocyte, in case you forgot, as well as LH, luteinizing hormone. All right, so then we get to what we call an antral follicle. We call it an antral follicle because we have this fluid-filled antrum now. All right, so now we have this fluid-filled antrum. also surrounded by granulosa cells. Notice that when an ovary is stimulated to develop follicles, only one ovary at a time is going to do it, and two, 6 to 12 primary follicles are going to start the development process. All right, so it's not like you're getting one that's going. You're getting 6 to 12 at a time. Only one reaches a full maturation cycle. What happens to the rest? They degrade. All right, when you get to this antral phase, we start to get a little bit different structure. We have these thecal cells. We have a theca interna, and these are surrounding the granulosa cells, and a theca externa. The theca interna, internal. So think of it as a double-layered membrane. All right, not a cell membrane, a membrane surrounding the follicle. The theca interna, the inner membrane, secretes estrogen and progesterone, our female sex steroids. And the externa is a vascular capsule, all right, more of a protective function than anything. All right, so theca externa, theca interna. The green are granulosa cells. Here's your oocyte. Here's that fluid. All right, as it's further developing, notice, I always say, here's what it looks like. I always tell my undergrad students, it reminds me of that scene in Indiana Jones where you have this idol on top of a pedestal, right? So here's your idol, here's the pedestal. You know a follicle is fully mature when you see that structure. All right? So this is the rest of it. Here's the fluid. And notice that the fluid is full of estrogen. All right? And the purpose of that estrogen is to increase the number of FSH receptors on granulosa cells. What happens if you increase the number of FSH receptors on granulosa cells? They're more likely to respond to FSH. All right? FSH, all right, we're talking about FSH from the anterior pituitary, not the receptors, as well as estrogen, also increase the number of LH receptors. So when we get to this vesicular follicle, getting to the mature state, you're increasing the number of receptors on your granulosa cells. And that's important because these receptors are going to bind the hormone and cause big effects. Okay, and we'll see that later in a few minutes. All right, luteinizing hormone as well as estrogen again, so increase of proliferation and secretions from thecal cells. So LH increases the proliferation, secretion from thecal cells. And what's the secretion from thecal cells? Estrogens, right? So you're increasing the amount of estrogen. All right, how do we know? Here's your estrogen curve, and it's going up, right? So as you are getting more receptors on your cells, and those receptors are being stimulated to produce more hormone, that hormone then is going to have a feedback loop to produce even more hormones. So that's why your estrogen starts going way up, right? But if it's causing proliferation of thecal cells, that's going to increase your overall size of the follicle as well as the oocyte. So the oocyte is going to get really big. So you go from this really small primordial follicle to something 1,000 times the size. That's not the scale. All right. So let's break this down. you have a mature follicle, also known as a Graafian follicle. All right? It's going to be secreting estrogen. All right? As it's secreting estrogen, as estrogen is increasing, estrogen can go back to the anterior pituitary and inhibit follicle-stimulating hormone. All right? What happens when you inhibit follicle-stimulating hormone? You're not stimulating follicles to mature, right? So what happens is that all those 6 to 12, let's say 5 to 11 others, they stop maturing. And only one that's developed to the point where of no return goes on for ovulation. All right, so that increase in estrogen decreases FSH, which feeds back to the follicles that aren't as mature and prevents them from developing any further. Because how many oocytes do we want to release? 1. If we didn't have that happen, we'd be releasing 6 to 12 oocytes. Then what's going to happen? We're going to deliver puppies. Right? We're going to deliver multiple babies. We don't want that to happen.
What happens in the case of maternal twins? Like, is that just it happens too late and two follicles go through, or?
Yeah.
Okay.
Yep. So sometimes it breaks down, right? When women go on fertility medications, that will tend to have more, ovulate more oocytes, and that's why you have an increase in the number of births, babies per birth when you're on fertility meds, because it doesn't allow this to happen effectively. All right. So then that the one ring, will develop in a mature follicle and will be the only one that ovulates. By ovulation, we mean secreting that oocyte from the ovary. All right. The rest of these, what we call involution, they break down, they get resorbed. There's some evidence to suggest that they become like scar tissue within the ovary. And when you have enough scar tissue built up in the ovary, you start undergoing menopause. Okay? All right. So now we have this mature follicle, and it says, hey, I'm getting rid of you. Go off and do good things. Which is a big lie, right? Because 99% of the time they die. Right? So ovulation occurs. Now, ovulation occurs because we have a big release of luteinizing hormone. And you've probably heard of this before, what we call an LH surge. All right, so luteinizing hormone will spike all of a sudden. That will stimulate the release of follicle steroid hormones, mostly progesterone, right? Which is why you see progesterone start to come up in a big way right after ovulation. That's going to the progesterone's going to cause proteolytic enzymes to be released from the theca externa. That's going to weaken the follicle wall. All right, because remember, you're in a follicle still. So that luteinizing hormone is going to cause the wall of the theca externa, remember that's the protective layer, to weaken. And if it weakens, what can happen? You can push things out of it. All right, at the same time, it's going to have follicular hyperemia. What's hyperemia? Increased blood flow. Increased blood. And prostaglandin secretion, which will open up blood vessels, all right, which will allow more plasma to get into the follicle, So, okay, you've weakened the protective layer, and you're putting a bunch of fluid in there. What's happening? And when it gets too big, what's going to happen? It's going to burst. So literally, this LH surge weakens the wall, fills it up with fluid, and like a water balloon that's too full, it's going to burst. All right? So follicle rupture and release of that oocyte. Now, specifically, it's interesting to think about. I don't know how, in detail, the follicle orients itself so it doesn't burst into the ovary as opposed to out of the ovary. Like, I'm sure there's a molecular process that's going on that regulates that I don't know, so I can't give you that information. This isn't my wheelhouse. I mostly stay away from reproductive stuff. Okay? But in a direction, it's released, gets picked up by the fimbriae of the uterine tube, and then those cilia start to transport it. Okay? Now, what happens to the rest of this? Like, you still have that follicle left over. What happens to the rest of that? It becomes what we call the corpus luteum. Okay, we've talked about all that. It becomes the corpus luteum. And so then we enter what we call the luteal phase. And so we'll talk about what the corpus luteum is, what its purpose is, and what it's doing as we go along. But what's the purpose of the oocyte? Like, why does it get released?
To get fertilized.
Pardon?
To get fertilized.
To get fertilized, so we can reproduce our species. Right? That's why it does that. Now, this happens every month, right? Does it get fertilized? Yeah. Yes. I get it. No, my wife and I have had these discussions. I'm like, I, in no way, shape, or form, want to be a woman because I do not want to go through that. Like, I'm sorry. I do feel for you. Because it's not something I have any interest in doing.
I don't think we do either.
Yeah, I mean, to the point where, like, I would probably, like, have a hysterectomy and not have to deal with it ever. All right, so, but this is a guy talking too. Like, I would have probably different psychology if I were really a female, but. But if you know you never want kids, like, why not? I'm not promoting this, by the way. All right, so we have our primordial follicles, which have our primordial oocyte, the germ cell. And, at birth, you have 1 to 2 million oocytes. And by the time they get to birth, they've already undergone or started undergoing meiosis. All right, they've already started the process of division. And then each month, when that follicle is maturing and you get a secondary oocyte, it has completed the first meiotic division. And what happens is it's an unequal cell division. So you have this one oocyte, and it divides. One oocyte gives all of its cytoplasm to the other oocyte, leaving it with very little cytoplasm. That dies, and this goes on, which is very different than what we saw with males. For every one cell that goes into making sperm, how many sperm do we get? 4. We get 4 sperm. But for every oocyte that starts undergoing meiotic division, how many egg cells do we get? How many oocytes do we get at the end? Just 1. Just 1. All right? So, it goes there. It's going to stay. It's not going to undergo its second meiotic division until it gets fertilized. So most of our oocytes that get released every single month get released with only a single meiotic division. They never undergo that second meiotic division. That only happens upon fertilization. Okay, and again, when fertilization occurs, all the cytoplasm goes to one oocyte, and you just get the one. All right, which to me, kind of, if you're looking from a human variability perspective, you're reducing the total amount of variability you can have, right, by doing that. Okay, so if it doesn't get fertilized, however, what happens is that it goes out with the rest during menstruation. All right. Then in the ovary, you have this corpus luteum being generated. Okay. Spermatogenesis versus oogenesis. I think I talked about all of that. So I'm not going. That's just words that I've already said. All right. With female sex hormones, same pathway as males. We have the HPG axis. Only instead of testes, it's the ovaries. G stands for gonad. So GnRH from the hypothalamus goes to the anterior pituitary, causes release of FSH and LH, which go to the ovaries and cause the follicles to develop if it's FSH, and causes hormone secretion, as we just talked about in those previous several slides, if it's LH, okay, as well as ovulation. All right, but then these sex steroids, so this is a normal pathway, and this is happening all the time in the background. Okay, what we just talked about is specifically related to the menstrual cycle, but we also have to make sure that these estrogens and progesterones are causing breasts to develop, causing secondary female sex characteristics, so that they can prepare the female body for giving birth. All right. Luteal phase. Okay, it's called the luteal phase because you've gotten rid of the oocyte, and what's left over is the corpus luteum, hence luteal phase. Does anybody remember from when you've had this before what luteal means? Pardon? I didn't hear you. Nope. Yellow body, corpus luteum, yellow body. All right, we've released the oocyte with that LH surge. Now that we have this corpus luteum, that's going to actually be what's left over from the granulosa cells and the theca interna, because remember, we destroyed the theca externa. Luteinizing hormone is going to, that from the spike, is going to stimulate the formation of the corpus luteum, and notice that that's going to grow until about 7 to 8 days after ovulation. So after ovulation, that corpus luteum is going to get bigger and bigger and bigger as a result of that huge LH surge. Right? And remember, we have a lot of LH receptors on these cells too, so that's going to be part of why that happened. All right, so the corpus luteum has granulosa cells. Most of it is granulosa cells. 20% is theca interna. Granulosa cells, a lot of endoplasmic reticulum, a lot of aromatase, and aromatase is involved in making sex steroids. So you're going to produce a lot of estrogen and a lot more progesterone. So in the luteal phase, that's why you see estrogen, that's red, increasing, but a lot more progesterone increasing. All right? And that increase in estrogen and progesterone is going to go back to the anterior pituitary, shut off FSH and LH, and that's why you see this drop right here. Because that corpus luteum is secreting a lot of female sex steroids, causing LH and FSH to drop. All right? Okay, it's going to last for 7 to 8 days. If the oocyte is not fertilized in that 7 to 8 days, it starts to break down. It involutes. That means it folds in on itself, loses the capacity to secrete steroid hormones, and becomes the white body, or corpus albicans. And this is the other part. Like, once this forms, it's a lot bigger structure than all the other follicles that we're developing. So this is what, like that scar tissue that I mentioned, this is primarily where that would come from. So this picture is showing you the different picture of the same thing that was on the previous cell. What I didn't mention is that thecal cells will produce androgens. Those thecal cells will secrete those androgens to the granulosa cells, and because granulosa cells have aromatase, you're going to produce more estrogens from that. All right, so androgens will be put into those granulosa cells so you can make more female steroid hormones, specifically estrogen.